A very good morning to all. On behalf of Aditya Bangalore Institute of Pharmacy Education and Research, I welcome all the participants. Myself, Narahari, Assistant Professor, Department of Pharmaceutics. Today, the webinar is on chiral or the chiral compounds, we call it as. So chemical compounds that come as mirror image pairs are sometimes compared with the left hand and right hand gloves. And they are referred to by chemists as chiral or handed molecules. Each twin is called an enantiomer. Scientists believe that mirror image chemistry may also shed light on the nature and origin of life itself. On Earth, at least the building blocks of life are mostly left-handed molecules rather than the random mixture of left-handed and right-handed molecules found in extraterrestrial objects like meteorites. Many chemicals which are important to life are combinations of mirror image twins in which one partner may cure disease, quell headaches, or smell good, while its mirror reversed counterpart may be poisonous, smell repugnant, or simply be inert. Scientists are investigating the chemistry of thousands of such pairs, learning to make pure forms of the desirable twins and thereby creating many new products. Despite the close resemblance of such twins, the differences in their biological properties can be profound. Because they behave very differently from each other in the body, chiral twins are coming under increasing scrutiny among chemical and pharmaceutical manufacturers, biologists, agronomists, entomologists, and also regulatory agencies. So there has been an enormous recent progress in asymmetric synthesis. To enlighten us more, Aditya Bangalore Institute of Pharmacy Education and Research, Bangalore, in association with IPA, has organized this webinar on the topic, "Let us explore the science of mirror image molecules." The resource person today is a retired professor of Quality Assurance, Department of Pharmacy, Annamalai University, Tamil Nadu. He has over 30 years of rich experience in teaching and research. Since 2018 September, he is an adjunct professor at IISM SRM Institute of Science and Technology, Kattankulathur. His mission has been to spread the importance of chirality in pharmaceutical R&D and invite more researchers into the chiral research landscape. He has been the visiting professor to Philadelphia College of Pharmacy, University of Sciences in Philadelphia, USA, to pursue studies on chiral compounds in the year 2008. He has also been visiting professor of Indian Society of Technical Education in Pharmacy. He has been invited as speaker at various international and national conferences, symposium or congress, including Indian Pharmaceutical Congress, APTI National Convention, and Chiral India. So he has successfully completed number of research projects which are funded by government agencies like DST. He was the coordinator of UGC Phase 2 project with a funding of 1.2 crores, thrust area being chiral and nanotechnology. So he has also been involved in total research papers has been more than 60 with, and he has supervised many PhD students. And he has also awarded 11 MPharm theses and also supervised around 67. So editorial contributions, he has many, like he has been a member of editorial board of 3 different boards, and he has been professional reviewer of analytical and bioanalytical chemistry in Springer, Journal of Pharmaceutical and Biomedical Analysis, Elsevier, Journal of Separation Science, Willy, Chromatographia, Springer, and then Journal of Chromatographic Science, Oxford, Journal of Pharmacy and Pharmaceutical Sciences, and many more. So he has also been affiliated member and of the Management Committee of Chromatographic Society of India, we call it as CSI, and he has also been the associate editor of the CSI newsletter for the period 2010 to 2013. So I think he has been the most established academician. So we welcome this humongous academician, Dr. Valliappan K sir, who is the retired professor at Annamalai University, Tamil Nadu. I welcome you on behalf of Abhaypa sir. So I would like to request Dr. Valliappan sir to take over the session, please.
That's fine. Thank you so much, Dr. Narahari. Yes?
Thank you. Thank you, sir.
Nice introduction. Thank you so much. Good morning, everybody. Am I audible?
Yes, yes, you are audible, sir. You are audible.
I'll switch off my video as well, okay?
Yeah, that's fine. That's fine. Yeah.
So I deem it an honor to associate myself with the webinar organized by Aditya Bangalore Institute of Pharmacy Education and Research. Let me thank Dr. Sujitra, the principal, then Dr. Narihari and others involved in the conduct of this webinar for this opportunity. During the last 30 years of my mission has been to spread the prominence of chirality in pharmaceutical research and also to invite more researchers into chiral landscape. During this long journey, I did observe a phenomenon called stereophobia that exists among both students and teachers. When I mean stereophobia, I mean an allergy to stereochemistry or a hatred to stereochemistry, a fear to stereochemistry. People, even for instance, even today this seminar, when they're announcing possibly, many of the teachers and students might be thinking, this is stereochemistry. I'm not related to that. It isn't that. This science comes into play if the molecule under question is chiral. If a molecule is stereoisomeric, this science comes into picture. So it's become a very important science, and this lecture basically is an invitation kind of lecture. Nothing serious will happen here, and I'm trying to introduce you to the chiral landscape, and let's go into the chiral world. I understand the time slot is 60 minutes. I believe I should be able to get it done on time. Let's get going, and with this note, let me get to do my presentation on let us explore the science of mirror image molecules. So this is the purpose of the presentation. I'll introduce you to the chiral world, highlight prominence of chirality, need for mirror image perspective in drug research, regulatory stand on chiral issues, rediscovery of stereochemistry, why and its impact, and research opportunities it throws open. So this is the purpose of the presentation, and this is a sketch we'll build up as we go through each menu and then build up the presentation. Before getting the presentation, my customary that I normally get into two of my favorite quotes by one of the pioneering stereochemists, Louis Pasteur, who went on to say that universe is chiral. So this statement says so much about the prominence of chirality. He says universe is chiral. And the statement is, it says the sad truth is that we still learn most of our chemistry in flat land to the detriment of our science, meaning that, we do most of our chemistry in two-dimensional, whereas the environment in which we are working is three-dimensional. So if the environment is three-dimensional, if your molecule is three-dimensional, then your study need to be from a three-dimensional perspective. So he says the need for a mirror image perspective. So this code, let me get into first slide on chirality. This is most of you would have done in undergraduate classes. If I have an object and there's a mirror image, if I cannot superimpose, I refer such pair as enantiomers. You call them as mirror image molecules. You call them as enantiomers or handed molecules, asymmetric molecules, chiral molecules, so on and so forth. The requirement is the object should not be able to superimpose on its mirror image. So any object without symmetry, you may put them as chiral. And so just before getting to the further, let me take you to the world of symmetry. You find here, all these insects, creatures have a vertical axis of symmetry. Butterfly, the spider here, or the dragonfly, the deer, all of them has got a vertical axis of symmetry. The tiger is a vertical axis of symmetry. Beautiful butterfly, symmetry element on the center. It's a vertical axis of symmetry. When you go around the beach, you find a lot of symmetry in the crabs and spiders, what you find on the beach. But of course, you may not find time to see all that, but maybe when you go next time, take a look at these beautiful creatures. Then morphologically, human face, there's a vertical axis of symmetry. Of course, architectural marvel, Taj Mahal, symmetry is a thing of beauty. You find that there's a perfect symmetry out here on this man-made marble. Here you find a reflection symmetry on the water. Of course, this is very important. And this is the Twin Towers in Kuala Lumpur. This structure itself has got a vertical axis of symmetry. So symmetry is a thing of beauty. But we are talking about molecules which are not symmetric. Getting into the chiral concepts. This is a racemic pair. I call them as an enantiomeric pair. This is R is indicating the absolute configuration. This is a mirror image is S. So it's a pair here. This is racemic mixture, 50-50 equimolecular mixture of the enantiomers. You find here 6 of them towards the left and 6 hands towards the right. So it's a equimolecular mixture. You call them as racemic mixture, sometimes racemates. They are basically optically inactive. This slide shows an enantiomically pure molecule, enantiopure you may call it. Optically pure term is used earlier, but today this term is obsolete because early days The only tool available to measure optical activity was based on optical principles. But today we've got a lot of chromatographic tools. So we use the term enantiomer or enantiomerically pure, which means that all of them towards wind direction. There is no impurity at all. All of them having same configuration. This slide showed you enantiomerically excess or enantiomerically enriched. You find here 8 of them towards the left and 1 other towards the this is an impurity you might assume as an impurity. So this is called as enantiomeric excess, like 99.9%. That's what you're looking for when you synthesize a chiral molecule. This is a simple relationship which you do in your undergraduate classes with more than one chiral center. SS, the mirror image is RR, and this is SS, this is RR. These are related as diastereoisomers. This can be, maybe you will appreciate it better on this. This is a mesotartaric acid. It's a total reflection. So these two are related as 1 and 2 as enantiomers, and this is a meso compound. 1 and 3 are related as diastereoisomers. So having said that, this is a very important, tool which we use and the parameter which we study to assess chiral purity. Enantiomeric purity is usually reported in terms of enantiomeric excess, and when you perform a chromatographic separations, if you have two enantiomers, you get 2 peaks. You find here both of them have got same areas, 100, so the enantiomeric excess is 0 here. Here you find enantiomeric excess is 80%. One is 90, the other smaller peak is 10%. You find here this is 98, this is 1, and the enantiomeric excess is 98%. So this is a formula which is employed. Major peak minus minor upon major plus minor into 100, and you normally look for an enantiomeric purity of 99.9% if you are synthesizing a pure enantiomer or a drug molecule. So this is normally used, and the tools employed are to measure enantiomeric purity. There are a wide range of tools. Polarimetry is there, CD spectroscopy is there, NMR, chromatography, but most popular tool is chromatography. Okay, having said that, now we go into chiral technology, exploration, and understanding. We'll try to reflect upon the prominence and the need for chiral perspective from these menus. We'll go into each of these menus and try to justify the prominence and the need for chiral perspective in your research work. Let's go to nature. Very clearly, nature is not even-handed. Nature is very stereospecific. I'll give you 1 or 2 examples here, which you are all familiar, most of you are students, and I understand these molecules are very familiar. These are millennium-old drugs. One is morphine, other one is quinine. And you look at morphine. Morphine is a very highly demanding chemical structure with 5 stereogenic centers or 5 chiral centers. When I mean 5 chiral centers, the molecule can exist in 2 to the power of 5 possible stereoisomers, which means 32 stereoisomers. So morphine can exist in 32 different stereoisomeric forms. Among that, only the LIVO form having this configuration, 5R, 6S, 3R, 13S, 14R have got the desired biological activity of narcotic analgesic. So levomorphine has got the desired activity, and that's the only isomer or enantiomer synthesized by poppy plant or Papaver somniferum. So nature is very stereospecific. If human being we have to synthesize, we need to look for very sophisticated synthesis. We name them as chiral synthesis. You need very, very advanced research facilities and compounds, and you might get a Nobel Prize. People have got Nobel Prize for that. But nature does it just like that. That's the beauty of nature. And if you look at this molecule, quinine, it's an anti-malarial agent. Here again, you find this molecule has got 4 stereogenic centers, 1, 2, 3, and 4. And if you look at that, 2 to the power meaning which has got, I think 16, 2 into 2, 8 into 2, 16, 32, 2 to the power of 4, 16 stereoisomer, I'm putting mathematics. You find that in that, only the livoquinine has got the desired anti-malarial activity, which means that the nature is very stereospecific, and the hallmark of nature, molecules in nature is a unique chiral or homochiral, you may call them. They exist in one of the forms. You don't find, but the moment we synthesize molecules, you end up in racemic mixtures. That's so to get enantiomer selective molecules, you need chiral synthesis. We'll see that later. So nature is specific, and if you look at even now, the molecules, for example, all the amino acid residues that goes in the building up of natural proteins have got L configuration. Most of the sugars that goes into the building up of nucleic acids like RNA, DNA, all of them got D configurations. You find that essentially physiological processes are homochiral or unichiral, and they are 10% stereoselect. So that becomes very, very. So we find that nature is highly stereospecific, and to understand, you need a chiral analytical tool. Just look at bioenvironments. It's a very interesting thing, and you find that the behavior of a chiral molecule depends upon the environment in which it is present. Similar to our behavior. Our behavior depends upon the environment in which you are present. For example, you are on a webinar, you have a different behavior pattern. But you are on a live seminar, your behavior is different. You go to a theater with your parents, your behavior is different. You go to a theater with your girlfriend, the behavior is different. So your behavior is governed by the environment in which you are present. For a chiral molecule, there are only two environments. Either the environment can be chiral or the environment can be achiral, only two environments. But the behavior is pronounced only in a chiral environment. I'll give you a simple example to make this point clear. Let us look at the molecule. This is an ibuprofen, non-steroidal anti-inflammatory agent, which has got one chiral center. So the molecule can exist in enantiomeric pair. This is R and this is S. Normally, this in undergraduate classes, this has got a carboxylic function. So you estimate it using an alkali. You titrate with an alkali sodium hydroxide, you get the total content. By alkalimetry, you get the total content, but you will not know how much of R is present and how much of S is present. In other words, you will not know how much left-handed molecule is present and how much of right-handed molecule is present. So to understand that, you need to have a chiral environment. Even if you perform an HPLC method using ODS column, which is in a chiral environment, you cannot differentiate how much R is present, how much S is present. So to understand that, you need to make the environment chiral, and then you'll be able to quantify how much R is present and how much S is present. So it all depends upon the environment in which you're working. But interestingly, biological systems and human body are at the molecular level are highly intensely chiral. We are filled with chiral discriminators like amino acids, proteins, carbohydrates. All these are chiral. So human system can, or biological system can, differentiate left-handed molecules from right-handed molecules. In other words, if I place a racemic molecule into a biological system, it will act upon them differently. Your metabolism will be different, elimination will be different. So you have very complex situations out there. So I'll give you one simple example to make this point more clear. This is a simple molecule, limonene here. It's got a one chiral center, so the molecule can exist in two forms. One is called the R-limonene, other one is called the S-limonene. These are called as mirrored configuration. And you find that R-limonene has got an orange smell and S-limonene has got a lemon smell. The difference between these two is I'm changing the configuration. So when I change the configuration, I'm changing the shape of the molecule. Simple. When I go from R to S, what's happening is the shape is changing. And it's believed that because of one shape, it's able to trigger receptor site in the, it's an olfactory receptor in the nasal tract, and it's able to trigger and give you an orange smell. Another shape is triggering a receptor which is responsible for the lemon smell. So a simple change in the shape of the molecule is giving you a widely different biological perspective. In this case, it's an olfactory perspective. So think about drug molecules. I move from one configuration to another configuration. I may end up in a big spectrum of activities are possible, and I'll give some examples a little later as you go further. So the shape becomes very, very important, and this can be, I moved it 1 minute, excuse me. I moved the slide, and I'm sorry, 1 minute, I'll get to that. Okay, this can be explained using a simple, chemically you can explain like this, and if you assume the body configurations are, it is interacting with the enantiomeric pair. Drug R is this one enantiomer, this is the mirror image. Yes, it forms a complex RR, and this forms a complex RS. And as you're aware, these two are related as diastereoisomers because there's no complete inversion. This is RR, this is RS. So this is a diastereomeric complex. And as you know, they have different energy profiles and different physicochemical properties. And because of that, possibly, these enantiomers exhibit different biological properties. This can be a little more easily explained using a diagrammatic way. This is called Eisenstaedtman model, normally used in pharmacology. You find here, it's a drug molecule with a chiral center and three groups. These ligands are believed to attach to interact with the receptors. Here you find there's only 2-point interaction, 1-point interaction, single interaction. These two are not interacting. Here you find there's all 3-point interaction. You call this as a good fit, and you call this as eutomer in terms of in chiral chemistry. This is called distomer. Eutomer is the enantiomer with a desired biological activity. If in an enantiomeric pair, the activity which you're looking for, that enantiomer is called eutomer. The other one could be impurity is called as distomer. So this is another way of looking at it. And having said that, now just look at the kind of drugs available in the market. These are if you look at that, major proportion of the medicine in the market are chiral, and these drugs are believed to interact with biological systems. So biological systems are already chiral. If you look at this, you find that almost 67% of the medicine in the market today are chiral. This is a list from approved drugs between 2004 and 2006. Of course, even recent data will be telling you more. You find here 47% are enantiomer multi-chiral centers, 5% racemic, 18% enantiomeric single chiral center, and the rest, of course, multi-diastereoisomers. And you find major proportions, around 67% of the drug in the market today are chiral. So you find that the two partners of drug therapy, meaning biological systems or the human body and the drugs or of the drug therapy are chiral. So when two players are chiral, you can expect unexpected things, and we'll go with more examples to illustrate that. Having said that, now we'll go into the human system. How does it handle chiral events? This is a very, very popular, scheme which explains the biological phases of biological action. It's a drug, racemic drug, gets into the pharmaceutical phase. Then what there you have disintegration, dissolution happening, and what survives after that is called the pharmaceutical availability. Then it gets into the pharmacokinetic phase where you've got absorption, distribution, metabolism, and elimination. And what survives after that is called bioavailability or biological availability. Then it gets into the pharmacodynamic phase where you've got the ligand and protein or drug receptor interactions, and you get a response. The response could be an effect or a toxic, depending on the effect you look for. So you find here, racemic therapeutic by definition, it's 50-50. Racemic drug means 50%, 50% of the enantiomers. But the moment you place a racemic drug in a biological system, it is no more 50-50 because, you don't know how the metabolism will work. One will be metabolized, other will not be metabolized. So there will be always, you can expect 70-28, you don't know, 80-20, 70-30, 60-40. So you need to track individually to know how much in vivo it's available. So in the pharmacokinetic phase or pharmacodynamic phase, you need to track the enantiomers enantiomerically, and you need a chiral analytical tool. I'll give you a few examples to make this point clear. Look at the pharmacokinetic phase. It's a simple example here. Salbutamol is a bronchodilator, which is one chiral sender. So this molecule can exist in a pair of enantiomers. You find here the R enantiomer, the iotamer, undergoes faster metabolism than the inactive form. So what will happen is the active form is lost quickly, and so the bioavailability, there's a drop in the bioavailability. So to understand these kind of transformation, you need to perform an in vivo enantioselective pharmacokinetic studies. Whenever there's a possibility for stereoselective metabolism, then you need to have an enantioselective kinetic study. Otherwise, this data will not be available to you. Another example here. Look at this molecule. This is a dexrabiprofol and this is a levorabiprazole. These two are in anti-ulcer drug. You find here, all the parameters are different here. Cmax, for example, for the R dexrabiprozole is 576. You find here 234. Area under the curve, if it's 1972 here, it is 460. T half is 2.74, is 1.5. You find that all the kinetic parameters are differential properties, and so to understand, you need an enantioselective study. Otherwise, if you don't have a chiral perspective, you will miss the data, and whatever you do will be of non-science. One more example, of course, I did do that. Okay, fine. We'll go into the pharmacodynamic. I'll give a few examples, ample examples. I'll give one or two. Look at, for example, ibuprofen. Ibuprofen is, the iotamer is the yes enantiomer is 100 times more active than the distomer. Distomer is less active. Penicillamine is, it's used to treat Wilson's disease. The S form is used for treating Wilson's disease, whereas the R form has got a side effect like granulocytosis. Propoxyphene we'll discuss a little later. These are a pair of enantiomers. One has got analgesic activity, other one has got antitussive activity. More examples here. Propranolol. For example, yes enantiomer is 40 to 100 times more active than the distomer. Teramizole is an anthelmintic agent, and you find here this is a nematocide. This has got the desired activity, whereas the R form harbors a toxic effect. So you find that various possibilities are out here. Thyroxine. The S form has got the prescribed thyroid deficiency, activity able to control that, whereas you find the R has got able to decrease serum cholesterol, different biological activity. So you find wide spectrum of activities are possible between the enantiomers, and so there's a need to understand them, whether it's pharmacokinetic study or pharmacodynamic study or toxicological study. You need to have a mirror image perspective. Your study need to be enantioselective, otherwise these data will be of no use. All this applies if your molecule under question is chiral. Of course, you look at the toxicity. These are the drugs which are withdrawn from the world market because of stereochemically engineered toxicity. All these drugs are withdrawn from the world market because of stereochemically engineered toxicity. Terodolin, and derivalol, prelamine, bifenadine, all these are withdrawn from the market from stereochemically induced toxicity. And of course, thalidomide, will come to that a little later. This is another classical drug which has been responsible for the rediscovery of stereochemistry. Having said that, now let's look at the prominence in chiral technology. You find that in 2001, 3 people were given Nobel Prize in their work on chiral technology. Basically, they developed chiral catalyst which can be applied for synthesis of pharmaceuticals, for example, Knowles. Okay, I don't have the link here. It's okay. But the point here is 3 people got Nobel Prize in 2001, indicating the prominence of the field. Of course, this slide shows the world global market for chiral products, and you find that the global market is believed to reach somewhere around 6.6 billion by 2022. Again, this slide shows the kind of revenue generated from chiral chemicals and by application projected between the period 2013 to 24 in US dollars in millions. You find here 2013 it is somewhere around 10,000 US million dollars, and you find it is believed to expected to almost double in 2024. It is going somewhere around 20,000 million US dollars. So there's a big jump, and so chiral technology becomes very, very hot. If you look at here, the top 10 drugs you find, 9 of them are chiral. All these drugs, atorvastatin, simvastatin, clopidogrel, amlodipine, salmeterol, all these of the top 10 drugs you find them, 9 of them are chiral. Of course, this slide we did show that, and 67% of the drug in the market are chiral. These are the Indian companies which are actively involved in chiral research. I just wanted to focus you on that, and they are actively engaged in chiral research. The leading companies, Yamcure from Pune, then of course you have Dr. Reddy's, then Cipla, Zydus Cadila, Torrent, Shasen, Sunfarm, all these are involved in chiral research, and they're working on synthesizing enantiopium molecules. So the field is pretty important. If you look at regulatory environments, there's a very favorable government regulations. They are pointing towards the production of enantiopium molecules. There's a growing prominence of single enantiomer drugs. Then there's a rising significance of chiral chromatography technology. So there's the regulations are very favorable for enantiopium drugs, and so the field becomes very, very important. And the regulatory guidelines across the globe strongly encourage the development of single isomers and discourage stereoisomeric forms. And the approval could not be granted for a drug containing more than one isomer unless the pharmacokinetic and pharmacodynamic properties of each could be described and, more importantly, justified. So to market a racemic therapeutic, you need to justify that these enantiomers are pretty safe. Otherwise, you need to submit data on both the enantiomers separately. In fact, US FDA, European Union, and then Drug Licensing Authority Japan demand for enantiomer-specific data on pharmacokinetic, pharmacodynamic, toxicologic profile of chiral drugs to assess the safety and efficacy. So for instance, I'll just show you one slide from Japanese licensing authority. Look at read the last paragraph here. When the drug concerned is a racemic body or racemic mixture, it is recommended to investigate the absorption, distribution, metabolism, and excretion of each optical isomer separately. Otherwise, the drug will not be given approval or cannot be further processed. So this is the current regulatory issues are supporting chiral enantiomer drugs. And if you look into chiral drug development, again, you find every stage you need enantiomer assessment. There's a need for chiral perspective, especially in chiral drug development. The quality control formulated chiral drugs to confirm that manufacturing processes induce no unacceptable change in the enantiomeric purity of the final product and their stereochemical integrity may be maintained. For the proposed shelf life, you need chiral analytical tool. Then to monitor outcome of chiral synthesis in which crucial stereo centers are created. So you need a chiral tool. To guarantee chiral identity, purity of active substances before each batch release in chemical development. Assess enantiomer specificity of pharmacokinetic, pharmacodynamic, toxicological activity profiles. So in all these cases, you need a chiral tool. That's very important. To monitor formation of a chiral metabolite. To detect potential configuration inversions, you need a chiral analytical tool. So my point here is, if the last maybe 15 minutes, if I'm able to convince you that the mirror image perspective is very important if the molecule under question is chiral. If you are not working with a chiral molecule, then of course you don't have to worry about that. But if the molecule is chiral, then you need to have a mirror image perspective and you need to go for chiral analytical tools. Okay. Now just before getting into that, now let's see handedness and a rediscovery, rediscovery of stereochemistry, because stereochemistry is not new to us, Stereochemistry is not as early as Louis Pasteur. Will come to that later. I'll just let you go through some of these titles. These are the publications that have appeared between 1984 and 2004 in the news magazines and the leading journals, showing there's a sudden spurt of rediscovery of stereochemistry. Let's look at those titles. Mason, 1984, The Left Hand of Nature, New Scientist, 1984. Through the Chemical Looking Glass, New Scientist, 1991. These titles are very, very interesting. All these are pertaining to enantiochemistry. For example, mirror image chemistry yielding to new products, 1991, New York Times. Looking Glass Chemistry, Science, 1992. Drug firms sort their left from their rights, 1993. Lateral thinking, you think in different directions, the Time magazines, 1993. Mirror Images, The Guardian, 1994. Science, Mystery of Mirror Images, 1995. Efficacy, on the other hand, Drug Discovery Today, 1998. Life Spawns from Left-Handed Molecules, 2003, United Press International. Then, of course, one more here, New Process Shifts Mirror Image Molecules. So the point is, all these were happening between the period 1984 and 2004. That time, there was a rediscovery of stereochemistry. So why there's a rediscovery of stereochemistry? Just try to understand that. So before going to the stereospecific awareness level, events and stimulants, what made the rediscovery of stereochemistry and attitude and change, I'll quickly go through chiral timeline because I find a lot of students are out there and I would love to show them all these big scientists who are responsible for the development of the science as we treat today, buyout. 1815, he discovered optical activity of rotation. That means stereochemistry we know as early as 1815. Pasteur, 1848, first achieved separation of enantiomers, physical separation under a microscope based on the morphology of the crystals. He did it after 30 years after the discovery of optical activity. He's a Louis Pasteur. Then, of course, the foundation was laid by the Van Hoff and Lebel. They went on to propose the sp3 carbon, the three-dimensional carbon, which is the foundation for the stereochemistry today. When we have 4 ligands attached to a carbon, you call them as a chiral carbon, and they can exist as enantiomers. So these two of them independently proposed a theory in 1974. One is a Dutch chemist, other is a French chemist. So the theory of molecular chirality principle was established 145 years back, but today we talk about stereochemistry. And 2001 Nobel Prize was given to stereochemistry. Fischer is a carbohydrate chemistry. He visualized the fit between enzyme and substrate in the lock and key mechanism. It's a carbohydrate chemistry, 1894. Then 1904, the Kelvin introduced the term chiral. First he introduced the term chiral in 1904. Then later in 1926 was Arthur Cushney from some pharmacologist, British pharmacologist, who was the first to offer a definite example to show the difference between enantiomers of a chiral molecule. This was done after 50 years after Van Hoff-Lebel theory of isomerism. He went on he was able to concrete examples to say that one enantiomer, the liver enantiomer, is much more active than the racemic form, or the liver adrenaline is more active than the racemic mixture. So that's a first example given to science. Then of course you have the helical structure of the DNA. This is all because of the chirality of the fundamental building molecules. You have a helical structure. Then of course the Mislow's book you must be having in the introductory to stereochemistry. He reinforced the term chirality in the literature. The moment it comes to literature, of course, then it comes to books, and then you people get to read, we teach you people. So he was responsible for that, Kurt Mislow. Then 1960s, of course, you got the prologue, Kahn-Ingold prologue rule. You call them as, R and its notation, the ZD notation. He got Nobel Prize in 1974 in chemistry, research into stereochemistry of alkaloids, antibiotics, and enzymes. So we know stereochemistry as early as 1966, but there is a rediscovery today during the period of the early 20s. Why is that? To understand, I'm not going to this because it'll be a little heavy. I don't want to do that. I'll get to the events and stimulants that really happen. Because of that, you have rediscovery of stereochemistry. That I believe there are 2 reasons basically. One is called stereochemically induced drug toxicity. Another one is a breakthrough landmark papers. These 2 were combinations which made scientists to think on stereochemistry and believe that enantiomers have different biological activities, and there's a need to have a mirror image perspective when you study these molecules. So I'll first go into the drug-induced toxicity. Thalidomide is a classical example. We call them as thalidomide tragedy. This molecule was introduced into the world market in 1956. This was given to pregnant women to control nausea, vomiting in the early trimesters, and they found that the babies born had a condition called as phocomelia. This is called drug-induced toxicity. This is phocomelia, conditions where babies are born deformed, like hands and limbs, very painful condition. These are called as thalidomide babies, and the drug is called a monster drug. Then in 1961, the drug was withdrawn from the world market because of this, and they studied and then found that the molecule has got one chiral center, so it exists in enantiomeric form. The R form has got the desired activity, the beneficial sedative effect, whereas the S form, the mirror image form, harbors the toxicity or embryotoxicity. So this made the way we look at chiral molecules, and people started thinking that each enantiomer should be viewed as 2 different chemical compounds. So thalidomide tragedy is one of the key reasons for the way we look at chiral molecules. Totally change happened. And along with that, I find that these papers were also very, very important by Arens, and he was very critical in really, making scientists across the globe to think on these. And he wrote a series of papers. I'll just go through those titles. References are available, but I don't think you may need all that at this stage. So all these are by Arens, and you try to understand the title. Stereochemistry, a basis of sophisticated nonsense in pharmacokinetics and clinical pharmacology. Implication of neglect of stereochemistry in pharmacokinetic and clinical pharmacology, 1987. That's the same period. Selectivity, a natural aspect of molecular biology, a blind spot in clinical pharmacology and pharmacokinetics. Stereochemistry of bioactives and abiotics, a pre-Pasteur attitude in medicinal chemistry, pharmacokinetics, and clinical pharmacology. Selectivity in pharmacodynamics, and he went on writing all these kind of papers, and finally he declared and advocated that a therapeutically non-active isomer distomer in a racemate should be treated as impurity. Suppose I got a racemic therapeutic, and one form is active, other is inactive, means 50% is inactive, and that's called as distomer. And he said that the distomer should be regarded as an impurity, and you cannot give such pair to human beings. So neglect of stereochemical aspects of drug action degrades pharmacokinetic studies to expensive, highly sophisticated nonsense. So he said 15% impurity is not acceptable, and racemate should be treated as fixed drug combination. So say, when I say fixed drug combination means, say, paracetamol, ibuprofen. You know these two are two different drugs. So when I say racemic ibuprofen, you believe that these are two different forms of the same drug, but it isn't that. You need to treat them as two different chemical entities, and you need to perform a say for both of them. Everything should be done separately for both the enantiomers. So he said racemates to be treated as fixed drug combinations, and today it is being done like that. So these two things are the main reasons for a rediscovery of stereochemistry. These drug tragedies, and of course the papers by landmark papers by Professor E.J. He written volumes of drug discovery, drug design books. I'm sure you all would have been, some of you will be aware of that. Having said that, now let's see what is the attitude change and impact on this. So this attitude change has a lot of impact, especially on the research and development, in the academia and regulatory affairs or control. And today, enantiomers are treated as two different compounds and not two forms of the same compound. That means each version of the chiral twin is essentially viewed as separate chemical species. That means you treat them as a fixed drug combination, or you treat them as a polypharmacy. It's a major impact. And the consequences, there are, after this, a lot of consequences happen, like birth of new journals, chirality, enantiomers, asymmetry, tetrahedral. These are specialized journals which publish only articles pertaining to stereochemistry and mirror image molecules. Then, of course, constitution of an stereoisomeric committee by the CDER, Center for Drug Efficacy Research by USFD, indicating the prominence of stereochemistry. Separate committee which looks into all the aspects of drugs, research in stereochemistry and especially chiral drugs. Then there's an exponential explosion of chiral technology, like chiral HPLC, chiral NMR, chiral TLC, chiral chromatography, chiral superfluid chromatography. All these are coming. So there's an explosion of technology. Then there's the emergence of separate field called chiral pharmacology. Then there's a formulation of a FDA policy statement for developing new stereoisomeric drugs in 1992. You can go to the website to understand that. So all these shows the prominence and the impact of the rediscovery. Chiral assays are being done. Then, of course, enantiomer selective synthesis, Nobel Prize has been given. Then new nomenclature systems to reflect stereochemical nature of medicines. This you must be familiar. Many are coming up on the market, like levofloxacin. You don't use RNS. Public should be very clear. When I say levofloxacin, I mean it's a pure enantiomer of ofloxacin. Levofloxacin, it's a chiral switch. Citrusin is a racemic drug. Levocitrusin is a enantiomer. It is called a chiral switch or racemic switch. Ketoprofen is a racemic drug. Dexketoprofen is a pure enantiomer. So these kind of nomenclatures are coming. Yes, omeprazole. Omeprazole is a racemic therapeutic, whereas yes, omeprazole is a pure enantiomer. So these are called as chiral switches, and there are certain limitations which will be addressed, I believe. So new nomenclature coming up. Then there are recommendations to include chapters on drug stereochemistry in books on pharmacology. There are changes in editorial policies of journals. Then, of course, the chiral switches I'll talk about this a little later. Okay. So ultimately, the impact is that the component enantiomers of racemic drug may differ widely in their pharmacokinetic, pharmacodynamic, and toxicological profiles. So there's a need to track profiles of both the enantiomers separately in a chiral drug development. So if you're developing a chiral molecule, so every stages the study need to be from a mirror image perspective. Both the enantiomers need to be taken up. Everything should be done separately. Even the bioequivalence study, pharmacokinetic study, or toxicological study should be done for both the enantiomers separately. And there's a lot of pressure from regulatory agencies like USFDA for the production of enantiopure chiral drugs. So there's a trend towards enantiopure drugs, and today market is flooded with a lot of unique chiral drugs. So I just skip this because it will be little more serious. I don't want to do that here at this presentation. So having said that, now let's look into chiral research. What's the basic objective of chiral research? By this, the rationale is that you get less complex and more selective pharmacological profile, potential for an improved therapeutic index by moving into a single enantiomer, less complex pharmacokinetic profiling. So two enantiomers, I would only a single drug, reduce the potential for complex drug interactions, less complex relationships between plasma concentration and effect. So these are the specific idea or rationale for developing chiral specific drugs. What are the achievements? I'll give you one example or maybe two examples here. It's a very interesting molecule. I skipped it earlier. Propoxyphene. Propoxyphene, you find this is a pair of enantiomers. This is a levo, this is a dextro, and this is called as dexpropoxyphene. Dexpropoxyphene. This is dexpropoxyphene. This is levopropoxyphene. And you find that these are separated and marketed as two different products. This levopropoxyphene has got an antitussive action, and this dexpropoxyphene has got an analgesic action. So enantiomers for two different, widely different therapeutic indications. They are separated and marketed by a company called Eli Lilly USA, and today is in the market. So interestingly, you see a pair of enantiomer, you separate them, market them for different therapeutic indications, and it's in the market. And interestingly, the inventors have named them as mirror images. This is called as Novrat. This is the mirror image Darwan. So this is a very classical example where there's a pharmacodynamic complexity and you have two products from the same single molecule. Another interesting example is these are called as chiral switches. Esomeprazole was introduced by AstraZeneca in 2000. The claim is that higher dose efficiency and proton pump inhibitor. So you call them as chiral switches. I switch from omeprazole to esomeprazole. What are chiral switches? Chiral switches are these are you take an approved claimed market racemic drug and switch to a single enantiomer for a improved therapeutic efficacy or a managing like patent extension. That's a different aspect. So from racemic switch to single for a improved therapeutic efficacy or decreasing toxicity and all that. You don't treat them as inventions, but you treat them as selection inventions. Okay. So this is how the principle is. I got a racemic drug here. I switch them. I got a pure enantiomer here. But to do that, there's a need to what is called as you carry out bridging studies and confirm the authorities that regulatory agencies that by switching, I'm not losing any beneficiary activities. So to switch, I'm not going to much detail into that. There's a separate topic. So you can switch like this. Only thing is you need to perform a study and submit and convince and then get it done. Okay. So there are many more switches in the market. These are the switches in the market. I'm just giving you a select list here. Esomeprazole, levofloxacin, levalbuterol is a bronchodilator, dexibuprofen, dexketoprofen. There's a big list is out there. And these are the other further list. The companies are listed in the market. Unique chiral interactions in India. All these are the drugs in the Indian market. These are therapeutic indications, and these are the companies. You find that MCure is the leading company who are dealing with this. 1 minute, excuse me. Yeah. So all these drugs are in the market. So there are people working on that. Now let's look at the opportunities. I think we'll be another 10 to 15 minutes. We should be able to closing down. I'm coming to the last area where what is the opportunity as a pharmaceutical scientist or academician? Where do I find the applications? Because people feel I'm in chemistry, so I don't have any such opportunities. You be any part of pharmaceutical sciences. You be a synthetic medicinal chemist. You be a pharmacy practice. You be pharmaceutics. You be a pharmacologist. You be analytical chemist. Even of course, naturally pharmacognosy. But the only requirement is if the molecule is chiral, there's an opening there. So the field is very fertile. I'll take you into the synthetic medicinal chemistry, what all openings they have. So you can develop synthesis of enantiomer pure drugs, synthesis chiral intermediates. I said earlier, you got Nobel Prize for this. These people got Nobel Prize for developing chiral catalyst, which are applied for the production of a lot of pharmaceutical drugs. Then develop chiral catalyst, develop membrane technology for production of enantiomer pure drugs. Stereoselective bioconversions. This is some studies are done mainly done in Institute of Science in Bangalore. Chiral switch strategy. You can move from one racemic to another. That can be taken up. Then if you're a pharmacologist, there is of course enantioselective pharmacological studies can be done, like pharmacodynamic and toxicological profiling of chiral drugs. Investigate and validate existing dose of chiral drugs. Sometimes, there's a clear example here. The oral dose of verapamil need to be larger than originally believed necessary based on plasma concentration. They found that, because the drug that reaches the bloodstream is enriched in the less potent enantiomer, whereas the utomer is metabolized. So you need to reschedule the dosage. So all this can be done. Pharmacy practice, if you selective drug interactions can be studied. Selective drug therapeutic monitoring, all these can be using, HPLC can be done. If you're analytical chemist, of course, a lot of, you have developed enantiomer assays, chiral impurity profiling, developing stability indicating assays for chiral drugs. Of course, chiral separations, there are huge gadgets available. I will not be going into those details. We'll be looking, basically the focus will be on separation science, and here again, all these are the available gadgets: chiral GC, superfluid chromatography, thin layer chromatography, capillary electrophoresis, high performance secret chromatography, and so on and so forth. A lot of drugs are available, tools are available, and in this most popular tool, just for information, is, if you look, this is a list of publications being done between 1964 and 2004, and you find that most of the publications, somewhere around 375 publications, this is the number of publications, is done by chiral separations by HPLC, then by blue is by GC, then capillary electrophoresis, and then capillary electrokinetic chromatography. So you find that the major tool employed here is HPLC, and I think most of these institutions have HPLC. So you need to have a chiral column to perform studies, and these are the scientists leading, in the field of chiral separations and chiral chromatography. He's from Japan, Okamoto, for a diacetyl company. He's a flux chiral columns, prickle from prickle columns, and Armstrong for cavity, cyclodextrin columns. So just to introduce you, these are the people. I'm trying to motivate you people. That's my interest here. Not going to further depth. Maybe in another seminar or sometime we get an opportunity, we can do that. And as you go to Sutex, you find ample opportunities here. Enantioselective bioequivalent studies of racemic formulations, evaluation of enantioselective release studies from formulations, suspensions, co-precipitates containing chiral excipients. Most of the excipients are chiral. Lactose, cellulose, HPMs, all these are chiral. So you have a chiral excipient, you've got a chiral drug, so there's a possibility for interactions. Then of course from dermal patches, enantioselective transport into skin from topical dosage containing racemic drugs. So you formulate a racemic drug, the dermal patch, and look for the chirioselective membrane transport can be done. Then of course the enantioselective permeability of racemic drugs from oral dosages with coated tablets. Enantioselective dissolution studies, you perform regular dissolution study, but you can perform, if it's a chiral drug, you need to know how much of R is released, how much of S is released. This can be done using UV. For a lower concentration, you can use chiral HPLC. Then stereoselective dispersion of sustained release microspheres of chiral drug enantiomers and bioactive concentrations. Finally, develop stereoselective drug delivery for solid dosage forms. This I have done my work on that. What you do here is you're developing a stereoselective drug delivery system. We don't separate enantiomers here. You take a racemic drug, you make a formulation with chiral excipients and make the condition suitable that one enantiomer is released into the system, whereas the other enantiomer is you hold it. One is you hold it, the other one is released. So you manipulate the condition such that the desired enantiomer is released, other one is bound to the matrix, so that there's a stereoselective drug delivery system. There's a lot of papers, people are doing a lot of research. You can search for stereoselective drug delivery system in Google, you'll find ample number of papers coming up. So a lot of opportunities for Sutex as well. So with this, I'll go into the wrap up. And today, chirality is an essential pharma research kit component, and I welcome you all to the chiral landscape to take a look at the potential research opportunities available. This is something like go to in a hotel. You go to a hotel, you are given a menu card. You go sit there and try to taste, and if you like it, you order further, or you get into that. Otherwise, you reject it. So you get into the chiral landscape, look at the opportunities, because I again will tell you, this chiral chemistry or chiral technology is a thrust area in UGC, in CSIR, in DST, in SAP, all the funding agencies. This is a thrust area. So there's a possibility that if you go with a good project, then you can get a good funding. And of course, you need funding to get a chiral column, or this expensive investigation a little, but you should be able to do that with a proper, because there are a lot of funding agencies available. So if the molecule under study is stereoisomeric, in particular chiral, then all the investigations need to be done from a mirror image perspective, else the data generated will be meaningless or nonsense. So if the molecule under question which you're working is chiral, then you need to have a mirror image perspective. Otherwise, the data you generate will be meaningless and nonsense. So we live in a three-dimensional world, and we should insist on a three-dimensional science. That's very important. So if you have any questions and inquiries, I'll be glad to answer that. And it's a pleasure talking to you all. I hope I have been able to convince you that this field is so important, and I wish you all start liking stereochemistry. Thanks for your time, and Kerala yours, Valliappan.
Yeah, it was a really good session, sir.
Little I rushed because I got to attend another session. Sorry.
Yeah. That's fine.
One of my colleagues is presenting, and I want to listen to that. I hope I'm in too fast for them, and I don't know how they liked it. Generally,
yeah. At least the thing is that we have realized that chirality is one of the important aspects of research. So to all the departments, not only it's for chemistry, but it's for different sectors.
My intention is only that.
Yeah. Sir, we have two questions. Can we have the question answer session?
Why not?
Yeah. So the first question is about the manufacturing cost of separating the mirror image, and also if funds are required for large scale animal testing of specific enantiomers. So is it feasible or it requires huge funding?
No. See, for example, funding will be available because, to analytical scale separations, what you need is an HPLC and a chiral column, right? Okay. Chiral stationary phase will be requiring you a few lakhs, 1 or 2 lakhs, and you can get 1 or 2. And sometimes if you're in contact with some industry, you get complementary chiral column. Used ones is okay. But enantiopure drugs are a little costly. You need to, because you need a reference standard to always work with HPLC. You can get it from Sigma, Rich or something. It might be expensive little. Funding will be sufficient. You propose a project and then, scaling up will be difficult. If you want to scale up, then, you need a method analytical scale. Then you might need a, kind of, flash operators. You need to, separate them analytical. You need analytical preparatory columns and all that.
Right.
That might be a little expensive, but you need to do that. You identify there are companies who will do it for you. There's a Tesla company. You give the molecule, they separate and give you the pure enantiomers.
Okay.
But it will not that expensive. Every science will be expensive, but then you should be able to apply for a good grant. And then you see, anomaly, I'm proud to say that, during my stay, I could get 1.2 crores.
Okay.
I moved it to phase 2, I was a coordinator as a team, but still, we make the key contributions.
Okay.
So chiral technology, nanotechnology were the thrust areas, and I could get some 12 chiral columns and each costing somewhere around 10, 15 lakhs, then other things and all that. So if there is a will, there's a way. And to learn swimming, you need to jump into swimming pool. So,
Right.
and you are in Bangalore, there are big companies out there. You can
Yeah.
contact them and, you can have a tie-up and slowly start working on that. I think that should be possible. If there's an interest, it will be possible.
Fine. And just last question. I think it's an interesting one. Like all living things seems to be exclusively left-handed. So why nature has a bias in chirality?
It's a big, there's a lot of, quantum, it's a big papers out there. It's a more of, they talk about energy, then thermodynamics and all that.
Yeah.
I'm not an expert in that, but they say it has happened just like that, not just like that.
Okay.
There's a lot of papers out there, pure. They talk about energy and it has happened and then it is there. There's no specific reason, but nature is like that.
Okay.
There are fundamental papers, which I think will be difficult for us to even understand because they talk about, the energy and thermodynamic principles on that. If you Google, you'll find a lot of papers on that. I wanted to give a link here. I thought it may be not necessary.
Right. So I think it's a huge topic and huge research subject, I believe. So
yeah, last 30 years I'm only talking on this.
Right.
This is normally an invitation kind of presentation, which I don't make it very serious, so that people should, I strongly believe there's a stereophobia both amongst teachers, and I believe they'll accept that.
Okay.
Even today, many of your faculty might be thinking, that attitude and an allergy to stereochemistry.
Fine.
Teachers don't teach well. I will strongly believe you like your subject if you like your teacher.
Exactly.
I have wonderful teachers in chemistry from my BSc to my masters, like Professor Harikishan Singh to Dr. Thambi in Bangalore to Professor Karvekar. Then you teachers make all the difference.
Exactly.
The students love the teachers. Then they like the subject. So it all depends how your teacher is able to motivate you. I believe this presentation. I don't know the feedback. You can tell me the feedback later. Whether I'm okay or I should improve further.
I think we have messages in chat box. It's always been an excellent session. So they're saying that, and some people want that slides to be shared. So I just said, so can
this sharing, this will be difficult because it will be random because hyperlinks I go in. If you are very serious, I can send a PDF to you. You can share to them.
Right. And
what this slide will be, it's not in order. You would have seen that. I jumped from one.
Correct. Yeah.
Hyperlinks are there. So even if the image you straight away send them, they will not be able to. It won't follow the track which they want to. If they're serious, you can. I suggest you send me a mail or something.
Definitely.
I'll make a PDF in the regular sequence for them, and then I can.
Definitely, sir. Definitely. Just one thing to add that the one beautiful thing in your slide is on the right hand side we can see your name and a mirror image of it. So that's really.
I will everywhere you'll find the title itself. You'll find a mirror image.
Right. Yeah.
I am a little stereo maniac. That is also not good. But I am like, it should be normal. Not this extreme nor that extreme. But I am this too much extreme. Everything I see on that I don't know. That's my weakness. Anyway, you observe that. That's okay.
It's just the nature that it's the strength as well as the weakness.
I can stop sharing now, right?
Yeah, sure, sir. So I just want to say that on behalf of Abhaypur, I express my heartfelt gratitude to you, sir, Dr.Valliappan K, sir, for an in-depth session, and my sincere thanks to our Honorable Chairman, Dr. Vishwanath, for referring such an eminent speaker, and also for his continuous guidance.
Yeah, I would like to add a point here. I forgot.
Yeah, sure, sir. Sure.
Of course, I know your person, management, Dr. Vishwanath, and say hello to him. And to what do you call that? To Dr. Pushpavali, my colleague,
Yeah, okay.
in fact, she only asked me first whether you will be interested
All right. All right.
your faculty. I don't know which department she is. I think.
Pharmacognosy, sir. Pharmacognosy.
Yeah, yeah. In fact, we studied together at Government College of Pharmacy in 1982.
Okay.
So suddenly we were back in touch, and we had a group. So she wanted to know whether I could talk to your. But I missed your principal. Srita is not out there.
No, I think she's online, but I don't think she's here. She's not here.
That's fine. That's a good take. Say hello to her also. Thank you so much.
Yeah, yeah, sure.
Can I leave now?
Just a second, sir. We'll just end it. We'll just end it. So, and also my most importantly, thanks to all the participants for their active involvement and patient listening. I have just put the feedback link. Okay, so we have just shared it in the chat box. You can just click it and you'll get the certificate. I think immediately you can get it. So just wait for 5 minutes after you just click it and fill it. And take care of one thing that you fill your email ID correctly. Otherwise, I don't think you'll receive the certificates. So just make sure you're typing the email ID correctly. Okay. So take care, be safe. So thank you so much, sir. Thank you for the wonderful.
You just through mail you give me a feedback of the participants, so I like to know how to improve myself.
Definitely, sir.
I believe every presentation you have something to improve upon.
Definitely.
So you give me a feedback. I should not think I'm pretty. Maybe I'm 30 years I'm doing this, but every time I do it fresh.
Right.
So this webinar is a new technology. Of course, I'm doing it every week, but still there's maybe a lot of things which people generally feel I'm a little fast, but somehow I'm a little fast.
No, it was wonderful. It was wonderful. It was smooth. It was smooth. It happened without a glitch. So I think,
thank you so much.
Well. Yeah.
Say hello to you. Thank you.
Thank you so much, sir. Thank you.
Thank you.
Hello. Hello.